Welcome everybody to the Physiological Society sponsored session. This is the physiology of obesity from mechanisms to medicine. My name is Joe and Pete is also here co-chairing the session with me today. This series really began back in 2018 and at the European Congress of Obesity, before heading to experimental biology and accumulating in a one-day symposium at Physiology 2019. Given the current situation, we decided to take this series online, and throughout the focus has always been the promotion of early career researchers to connect the next generation of leaders within the field. Now, before I hand over to Pete, who will introduce our speakers today, I just wanted to have a few housekeeping notes. The session is being recorded, so please bear that in mind. If you could ask your questions via the Q&A button, which appears at the bottom of your screen, we can then put these to the speakers after both talks today. And if you would like to submit your questions anonymously, please do. Otherwise, add your name and your institution, and we'll read that out as part of the question. I'd like to thank both of our speakers today and remind you that this session continues next week, again at Tuesday at 2 p.m. We will be exploring the link between the gut and the pancreas and how these hormones released from these organs can be used to treat type 2 diabetes. Thank you, Pete.
Thanks, Joe. So first speaker is June Corrigan, and she's a research assistant in Alex Banks' lab at Beth Israel Deaconess Medical Center, and she's due to start her master's degree soon. And today she'll be presenting work from her recently published eLife paper on the genetic and environmental factors that contribute to variability in energy metabolism. So over to you, June.
All right, hello everyone. So today I'm going to be presenting a talk entitled A Big Data Approach to Understanding Metabolic Rate and Response in Laboratory Mice. So just to start off with, maintaining a healthy body weight in a mouse or a human, of course, requires the body to balance energy intake and energy expenditure. The body, of course, converts the food that it consumes to energy through a process called metabolism. And genetic and environmental factors can affect this metabolic rate and energy balance, contributing to conditions like obesity. So how do we measure energy metabolism? Well, generally we measure it by these powerful but also sensitive indirect calorimetry devices. What these indirect calorimetry devices do is measure metabolic rates using gas sensors to capture rates of change in oxygen consumption and carbon dioxide production. And we can see that rate of oxygen consumption being plotted over time in this time plot right here. And as well as oxygen consumption and CO2 production, physical activity can also be monitored by recording infrared beam breaks or using receivers. And food intake can also be used by using mass balances. So, of course, weight gain will result when food intake is outpacing metabolic rate. But inversely, if food intake is falling below the metabolic rate, then weight loss will ensue. So, of course, it's also important to be able to compare the physiological data presented across papers studying metabolism. However, comparing results from published studies that use indirect calorimetry has been somewhat hampered by an inconsistent application of analysis or analytical techniques. So as we can see here, we see two time plots from two sets of mice. And looking at the time plot, you can see that it looks like these mice represented in orange have a higher general oxygen consumption, hourly oxygen consumption, than the mice represented in blue. And you can also see that represented as an average in this bar plot next to the time plot. But many, many indirect calorimetry studies are sometimes subjected to inappropriate normalization attempts. And by normalization, I mean that this rate of oxygen consumption, for example, which is here measured in mLs per hour, is further divided by the mass in kilograms of the mice being studied. So this becomes a problem when, especially in studies of obesity, metabolic rates of mice with different body compositions or different body compositions are subjected to these inappropriate normalization attempts. Many groups have agreed that the optimal way to interpret these plots uses the Ancova statistical analysis, which is an ANOVA with body mass or body composition as a covariate. So what you can see here is that looking at the Ancova analysis with oxygen consumption on the left axis and mass on the x-axis shows that in the first case, both groups are actually shown to have the same metabolic rate when body mass is the covariate. However, in the second example, one group actually has a greater metabolic rate, which we think of as the group effect, meaning one group does actually have a greater metabolic rate, irrespective of body mass. However, we can also see a third example where one group with obese mice, for example, has a lower metabolic rate when lean body mass is the covariate. So this is just an example showing that we really do need standardization of statistical analyses in indirect calorimetry. But of course, we also need a reference for the magnitude of any metabolic changes that we do observe following this analysis. So what we did is to establish the normal range of metabolic rate under standardized experimental conditions. We analyzed a data set from the IMPC, or the International Knockout Mouse Phenotyping Consortium, which is an ambitious large-scale project with metabolic data from more than 30,000 mice, which includes around 10,000 wild type and 20,000 knockout mice. And these mice were all about 11 weeks of age, about 70% were male. And in the data set that we used, we looked at the 24-hour averaged metabolic data from all of these 30,000 mice. And what's special about this IMPC data set is that it uses very highly similar indirect calorimetry protocols. And it's also from mice experimented on across 10 different sites in 8 separate countries. So what we did with this data is we plotted the change, we plotted the relationship, just as I showed 2 slides previous, of total mass to energy expenditure. In this case, we're looking at energy expenditure and kcals per hour, and what we can see is that across all the sites, the IMPC data does demonstrate a mass effect, meaning that the bigger male and female mice are expending more energy under standard housing conditions. However, it is becomes somewhat obvious when looking at this chart, or looking at this plot, that mice from these different laboratories across the world appear to convert food to energy at different rates. And just to emphasize, these 10 sites, each in the IMPC dataset, each reported between 240 and 1,300 male wild-type mice. And we actually observed more than a threefold difference in the metabolic rate slopes among sites, which translates to between 1.18 and 5.32 1.18 to 5.32 times 10 to the second kilojoules per hour versus gram body mass between sites, despite, as I mentioned, the highly standardized experimental protocols on mice of very similar ages and very similar diets. And of course, this makes our problem harder if we're trying to determine what is normal for mouse metabolism. Now, these discrepancies could be due to small differences between how mice are kept in different laboratories, even when all of these measures to ensure standardization are being practiced. For example, the temperatures of the cages the mice are kept in, or how active the mice might be, might differ depending on the laboratory. So to investigate these differences further, we examined which factors were influencing metabolic rate, including the mass of the mice, the temperature at which the experiments were conducted at, and as well as the sex of the mice. So the ambient temperature for these mouse experiments actually did vary by site. A range of the ambient temperatures were reported for experiments performed by 3 of the sites, which are the sites that I'm showing you here on this slide, which were located in Germany, Canada, and the UK. However, the other sites, the 7 other sites, reported just one single nominal temperature. For example, they reported that the temperature stayed at 21 degrees Celsius throughout the entire experiment, which of course likely does not reflect the accurate, daily variation in room temperature, nor what temperature it is actually in the indirect calorimetry cages that the mice are kept in. And that, we suspected, may have been a major source of unexplained variation. So we also examined the contribution of sex as another mass-dependent covariate. We actually found within the subset of about 2,000 males and 1,100 female mice, we found no significant difference between the energy expenditure rates of male and female mice. And we also, similarly, for these 3 institutions that did report accurate temperature readings, we did observe nearly identical slopes for energy expenditure versus mass at each of these 3 institutions. And to answer a question about what effects temperature may have had on energy expenditure, we found that mice maintained at warmer temperatures, which we classify as being between 22 and 29 degrees Celsius, we found that there were significantly lower energy expenditure rates than mice who are maintained at colder temperatures. And we found, we found that, 67% of this variation in energy expenditure can actually be explained by differences among these 3 institutions. And these differences would be in the ambient temperature of the mice, the body composition of the mice, and very much less so the sex of the mice. And we also were interested in seeing whether seasonality affected the metabolic rate of the mice, and we found that there was virtually no contribution of season, meaning that the time of year that the experiment was conducted on the amount of variation among institutions in energy expenditure. So to dive deeper into sex differences in metabolic rate, there is a long-held assumption in metabolic research that female mice are more variable in metabolic rate than males, but we found little evidence of that. At 7 of the IPC sites, both sexes were examined. So we are looking at data from those 7 sites. But the sample sizes, as mentioned previously, of the 2 sexes were generally unequal in those sites. There were about twice as many males as there were females. So we also examined the distribution of variance in the male and female mice in this modified QQ plot. So what we're looking at here is we see that the slope of these blue and pink lines are representing the theoretical standard deviation for the energy expenditure at each sex. And we can see that the lines are relatively parallel, indicating similar variability, while points on the extremes, which do not fall on these lines, are representing the variation from the normal distribution. And we can see that actually the overall, this representation is suggesting that female mice actually have a qualitatively better fit and lower overall variation. So all of our analysis up to this point was purely on wild-type mice to help quantify the magnitude by which institution is influencing, or excuse me, temperature, mass, and sex is influencing these institutional differences in metabolic rate purely among wild-type mice of all of the same genetic strain. However, of course, genetic variations can also impact this metabolic rate. One of the goals of the IMPC is to use mouse models to understand the contribution of genetic factors to phenotypic variation. So they have about 2,300 different knockout strains within their data set. And what we did is we wanted to examine the phenotype of all of these knockout strains. And we wanted to also find the strains with the greatest impact on metabolic rate. So what we did was we fitted the effects of mass and ambient temperature, which we already discovered to affect metabolic rate. So we fit the mean energy expenditure of each knockout strain to the wild type model, which we had already created, which included mass and temperature. And we plotted what greater metabolic effect was still unexplained by mass or temperature. And that is represented here by the residuals of energy expenditure. And we can see in this histogram that the residuals are pretty normally distributed around 0, suggesting that this model, which includes mass and temperature, is pretty appropriately fitted. So what we can do, given that the IMPC has these uniform phenotyping standards, we are able to have a great comparison of the relative phenotypic magnitude, meaning which knockout strains have the greatest effect on metabolic rate. Also to sort of validate this data treatment, what we did was we plotted the IMPC data for these residual values versus body mass And what we did in this left plot is we plotted 10 genotypes that were previously shown to affect body weight in mice. Now, these strains affect weight by different mechanisms, including increasing food intake, limiting metabolic rate, and affecting energy absorption. And we can see that at least some of these genes are on the extremes, meaning that they are multiple, at least 1 to 3 standard deviations away from what we are considering normal metabolic rate. And on this right plot, we plotted the human genetic loci, which were linked to increased risk for obesity and body weight through genome-wide association studies. And what we did is we plotted the strains in the IMPC that had a mapped obesity locus that had phenotypes plus or minus 1 standard deviation from the mean metabolic rate. And we found strains with lower metabolic rates that might predict obesity susceptibility. We also wanted to place these genetic, we also wanted to place these, the effects of these genetic differences on metabolic rate in comparison to other interventions that are known to or thought to affect metabolic rate. Because as you could see, the normal distribution of changes in energy expenditure, which are not accounted for by institution, mass, or temperature, is pretty narrow, with basically 2/3 of all strains, which is 1 standard deviation, having very modest differences of plus or minus about 0.16 kilojoules per hour. And so we wanted to place these into a physiological context, comparing the magnitude of examples of the effects of age, voluntary exercise, adrenergic activation by CL, and cage temperature on metabolism, again in wild type mice. And as you can see, especially in temperature, especially you can see, and as well as CL, you can see that the effects of these factors are much greater in magnitude than any factors that we observed purely based on genetic variation. Now, we also wanted to see whether the phenotypic differences that we observed in genetics could be observed reproducibly at multiple sites, because there were 6 strains which were examined in at least 4 locations, 4 of the IMPC locations, as well as the CDKL1 strain, which was studied at 2 of the IMPC sites, as well as in our own lab. So we integrated our results into that as well. So on the left, you see a plot of energy expenditure versus body mass for male mice at each of the sites. And what it does is it's plotting each individual mouse at each site, but it doesn't really convey the difference between the local wild type populations at each site, and therefore it's challenging to make these direct comparisons. So to provide context for the magnitude of these phenotypes, we did fit, again, mass and activity if it was available, and temperature into a multiple linear regression model for the wild type male mice at each site. And we calculated that deviation from the model at each site and plotted those residual values on the right-hand side that you can see here. And we saw that among these sites, the only strain with consistent phenotypic findings was the CDKL1 strain, where energy expenditure values similar to wild type were observed for mice at all 3 locations tested. Meanwhile, at most of the other sites, bidirectional phenotypes were observed among sites. So in conclusion, these results are suggesting that, of course, it is extremely imperative for any scientists that are using or studying indirect calorimetry and reporting the results. It's also essential for them to report factors such as room temperature, weight or body composition, or the animal's activity level in these metabolic studies. And this can help scientists compare results, repeat their experiments, and speed up and help research into mouse metabolism. But we're also unsure of all that we've been able to determine all of the factors that affect mouse metabolism. So further studies are absolutely needed to identify these factors. And I just want to, that's the end of my presentation, and I just want to thank everyone in the Banks lab, including Deepti and Yuchin, as well as everyone in the MMPC Energy Balance Working Group and Dr. Owen McInnes. Thank you.
Great, thanks for that, June. It's a fantastic talk. And so we'll move on now to the second speaker, which will be followed by a short 10-15 minute or so Q&A session. So next up we have Dr. Katherine Kentistou, who is part of the first MRC doctoral training program in precision medicine at the University of Edinburgh, and is now an MRC-funded research fellow there in Nick Morton's lab. Her research focus over the last 4 years has been identifying and characterizing novel gene drivers of human adiposity and body composition. Over to you, Katherine.
Thank you, Pete. Okay, so hi, thank you for tuning in. As Pete said, I'm Katherine Kentistou. I'm a postdoctoral fellow at the University of Edinburgh, and today I'll be talking about the work that I did during my PhD, which looked at the genetic underpinnings of adiposity and taking gene candidates from GWAS gene discovery to their functional characterization. I also wanted to make a quick disclaimer now that as all of this work is unpublished, I'll be omitting a few sensitive pieces of information. But if anything is unclear at all, please ask at the end or email me. A quick introduction. Obesity is an important public health concern, and more than 600 million people worldwide are obese. In 2017, 4 million deaths were attributable to carrying excess weight and caused by obesity's comorbidities such as heart disease, diabetes, and cancer. Although obesity is commonly defined by having a BMI above 30 kilos per meter squared, we also know that the way fat is distributed through the body can directly affect one's risk of developing these diseases. More specifically, adipose tissue distribution can generally be grouped into 2 types, pear-shaped or apple-shaped. For simplification, the former is when there is an excess of subcutaneous fat, and particularly on the lower half of the body, and the latter is when there is visceral fat accumulation, particularly around and inside the intra-abdominal organs. Apple-shaped obesity confers a much higher disease risk than pear-shaped obesity, and both of these types have been shown to be heritable. Thus, identifying the genetic contributors towards these body composition trends could lead us to disease preventative insights. So far, large-scale genome-wide dissociation studies, or GWAS for short, of obesity have focused on assaying simple and accessible anthropometric phenotypes, such as BMI, which don't really require specialist equipment to be measured. The problem is that BMI, for example, cannot differentiate between different body shapes, such as pear and apple shape. And thus, its discriminative capacities over adipose tissue distribution are very limited. However, large population cohorts that can be used in a GWAS context have now started performing whole body imaging on their participants. The cohort I have been working with is called UK Biobank and consists of 500,000 participants from all across the UK. For these participants, there are over 2,000 phenotypes that have been gathered, including physical measures, questionnaire data, and excuse me, and of course, genotype information. Sadly, at the time that this work took place in 2017, out of the 500,000 total participants, DEXA scans were only measured for 5,000. As opposed to MRIs or CT scans, DEXA scans offer a whole body quantification of fat, lean, and bone mass. They do so for 7 major body regions: the android region, the gynoid region, for the arms, the legs, the trunk, and the total body area, and also for visceral adipose tissue. You can see here 2 examples of DEXA scans of a pear-shaped individual on the left and an apple-shaped individual on the right, matched in every other aspect apart from their body shape. However, DEXA scans are very expensive, monetarily and also in terms of manpower, to carry out in such large numbers and large cohorts. So the UK biobank cohort to me looked something like this: very large overall, but the DEXA body composition phenotypes that I was interested in were only available for a very small subset of the cohort. However, anthropometric phenotypes were available for the entirety of the UK biobank cohort. Things like height, weight, BMI, waist and hip circumference, and also by electrical impedance analysis measures. While perhaps crude, all of these provide proxies of body composition and adipose distribution. Thus, I use them in a linear model framework to try and predict the DEXA body composition phenotypes onto the entire UK biobank cohort, and did so quite efficiently. Overall, the imputed DEXA phenotypes, which I'll now call IDEXA for imputed DEXA, predicted the measured DEXA ones with 81% accuracy. You can see here the predictions for fat mass in the android region, separately for men and women. So then I proceeded to perform GWAS on the IDEXA phenotypes for all 500,000 participants in UK Biobank, excluding the ones that were used in my prediction models. This resulted in a major increase in discovery power and thousands of genome-wide significant loci. After filtering these loci for novelty and for discoverability in other anthropometric phenotypes, I collaborated with other cohorts that had actual DEXA measured for their participants to try and replicate some of these novel loci. In doing so, we gathered the total DEXA replication cohort of 18,000 participants, in which I attempted to replicate all the loci that were sufficiently powered to be detectable in this much smaller sample size. Out of the 27 submitted SNPs, 6 replicated at an FTR of 10%, while 19 exerted directionally consistent effects between discovery and replication, validating the IDEXA approach. Out of these 6 replicated loci, today I'll be talking to you about one in particular, which for the purposes of this talk, we'll call the Mpx Locus. The first thing I did was to show that the Mpx gene was indeed the causal gene at the locus. I did so using the GTX gene expression data alongside my GWAS summary statistics and a tool called Summary Database Mendelian Randomization. In doing so, I showed that there is a decrease in leg fat mass and also a decrease in MMPX expression per copy of the effect allele. This would mean that the anticipated effect of a null mutation in this gene would be reduced adiposity. The Mpx gene itself codes for a peptidase of fibrillar collagen. The Mpx protein cleaves collagen fibers and facilitates their maturation and the formation of collagen polymers. MMP genes in general, which stands for matrix metalloproteinase, they act as modifiers of the extracellular matrix. And by altering the availability of extracellular matrix components, often in a tissue-specific way. And many of them also have well-established roles in developmental processes, such as body composition. So the hypothesis I formed around the MMPX GWAS signal was that MMPX can alter the availability of mature collagen fibers in an adipose tissue-specific manner during ECM organization, thereby regulating canonical adipogenesis. To try and understand how it might do so in an anatomically similar system, we obtained mice with no mutations in MMPX and sought to metabolically phenotype them under obesogenic conditions. For all of the following slides that talk about the mouse phenotyping studies, the mice will be portrayed like this. We'll have yellow for the wild types, half yellow and half green for the heterozygous animals, and green for the mice with 2 copies of the null mutation, so homozygous for the knockout. I worked with 2 experimental cohorts, one including 5 heterozygous animals and age-matched wild-type litter mates, and one including 8 homozygous mice with controls. And all of these were male mice. Both of these cohorts had their body composition and fasting glucose assessed at the baseline time point using time domain nuclear magnetic resonance, or TDNMR for short, and an oral glucose tolerance test accordingly. The mice were then exposed to a high-fat diet for 13 weeks, during which time I weighed them weekly, and after which I repeated the TDNMR and oral glucose tolerance test. I also performed these tests on the heterozygous cohort at the midway time point of the study, so at 6 weeks. For the homozygous cohort, I also did an indirect calorimetry test for a couple of days before the beginning and at the end of the high-fat diet treatment. And this mismatch in tests is purely because of licensing regulations. The first thing that we saw when looking at the weight gain of the two cohorts over the 13-week high-fat diet treatment was that both of these mouse cohorts were more resistant to weight gain than the wild-type controls. When looking at their body composition, the heterozygous animals had significantly less fat mass at the 6-week time point, while this genotype effect was partially maintained but lessened by the 13-week time point, as you can see here. Body composition for the heterozygous animals corroborates this attenuation. A similar trend is seen in the oral glucose tolerance tests. The mice are more euglycemic after a short course of the high-fat diet treatment, but the effect is attenuated after the full 13 weeks for both of the genotypes. Quite strikingly, the indirect calorimetry experiment showed that the homozygous null mice were hyperphagic and had higher energy expenditure both before and after the high-fat diet. When looking at their respiratory exchange ratio, which is an index of their carbohydrate versus lipid fuel usage, we saw that after the high-fat diet treatment, the null mice had a higher RER than the wild types, while both groups, as you can see in this middle plot here on the right, exhibited a flattening of their RER after the high-fat diet treatment, indicating a loss of metabolic flexibility coupled with obesity. All of these taken together indicate that the Mpx knockout animals have an increased metabolic rate. Finally, when looking at the postmortem, there were no overt differences at the tissue level for either cohort after the end of the 13 weeks of the high-fat diet, or any histomorphometric differences in the fat pads of the heterozygous animals that might help explain the altered homeostasis in the homozygous animals. Unfortunately, histology on the null mice is still being delayed due to work restrictions, but coupled with the shorter gut length that is shown here, I suspect an intrinsically greater reliance on carboxylation and perhaps a general inflammatory phenotype, but this remains to be seen. As for the next steps for this work, several associated loci from my IDEXA GWAS were near by other MMP genes. Each of these genes take part in extracellular matrix remodeling in a slightly different way. So I'm currently working on understanding if, how, and when each of these genes might exert an effect on adipose tissue expansion, both within physiological developmental processes and also in a pathogenic disease context. So to summarize this work, I started by imputing whole body DEX adiposity phenotypes for a small subgroup of UK biobank onto the rest of the cohort, doing so with 81% predictive efficacy. This means that my effective IDEXA GWAS sample size is over 300,000 participants, and that is 15 times the size of the largest published DEXA GWAS to date. After filtering the genome-wide significant signals for novelty and discoverability in other phenotypes, I submitted the 27 strongest GWAS signals for external replication. After assigning gene causality at one of these successfully replicated loci, I followed it up in a mouse model carrying null mutations in the GWAS causal gene. The Mpx knockout mice were resistant to weight gain on a high-fat diet and exhibited a generally improved metabolic profile after a short course on the high-fat diet, which was attenuated by longer exposure. They also exhibited a clearly altered energy homeostasis, the causes of which are currently unknown. However, this work provides proof of principle for identifying novel and validated gene drivers of adiposity using imputed imaging phenotypes, and will hopefully lead to more interesting biology soon. And with that, I'd like to thank my supervisors, Nick Morton, Peter Josh, and Jim Wilson, my teammates and collaborators for their help, as well as the MRC and the University of Edinburgh for funding me and this work. We'll now take questions, but I'm also happy to chat via email. I've attached my email address here. And thank you for listening. And thanks to the Physiological Society for having me today. Thank you.
Thank you, Catherine. It was a great talk. So we'll now go over to the Q&A. And we had a question for June initially from Phil Langton from Bristol, who asked, were you surprised at the magnitude of variation for mice of the same mass? It appeared to be a factor of at least 2, perhaps more. And were the mice from different strains in the chart displayed for differences in the 3 sites?
So I believe that you're asking about the magnitude of variation among different sites. If not, hopefully I can answer your question. So we were not, I mean, most of that variation among mice of the same mass was, sorry, excuse me. And you're talking about mice from different strains in the chart displayed for differences in 3 sites. So that plot with showing the mice from the UK, Germany, and Canada was purely the wild type mice. And you are right that there is some variation among mice of the same mass. But we weren't necessarily surprised by that, just because, as we say, there are so many environmental factors that do actually end up explaining, at least in these 3 sites, virtually all of that variation, including the differences in temperature, as well as the activity of the mice, and also in sites where we were able to look at, yes, activity, then that, which I believe is not in all these sites, but where activity was able to be studied, that also explained the bulk of that variation among the mice.
Thanks, June. So we had another couple of questions on, let me just find where it's gone on here. So one from Fatima from Liverpool John Moores, and I think a couple of people asked it. So did you record food intake? Was that comparable between sites? Was that a source of variation?
Food intake was only recorded at a smaller portion of the 10 sites. And unfortunately, there was a large amount of variability among food consumption, to enough to make us suspect that the way that the food consumption was recorded was less than precise, meaning not precise enough to warrant inclusion in our analysis. But definitely as methods to record food consumption, indirect calorimetry, which has historically been a source of a lot of unwanted variation, as that improves and as more experiments are recording that food consumption, that would definitely be something we would like to include in further analyses.
Okay, and I think following on from that, there was one about postprandial diet-induced thermogenesis. Do you think potentially that could be a factor in variability?
It definitely could. We did, thankfully, the IMPC did control at least for differences based on circadian rhythm. I believe all of the experiments, the light cycle for the mice started at 7 a.m. and the dark cycle started at 7 p.m. And we see generally that mice tend to eat basically as soon as the dark cycle begins, or even they're able to preempt that and start eating a bit earlier. So we're assuming that at least the mice were all eating at similar times of day.
Thanks, June. I was interested in whether a wild type mouse is a true C57 mouse brought in, or is this ones that have been bred in-house and therefore may have some genetic drift?
That is a good question. I believe that these mice were bred in-house, so there definitely is yes, there definitely is a possibility that genetic drift does have an effect. But again, we were not really able to investigate that.
Yeah. And I presume when these mice are in the metabolic cages, they're singly housed. Was that standardized also? Were they singly housed for a period before entering the cages, or were they group housed?
That is a question that I unfortunately am not sure of the answer to. So in our metabolic cage experiments, at least, we do, as you say, individually house the mice while they're in the calorimeter, because that's the only way that it will work. But we also we always try to incorporate an acclimation period of maybe 18 to 24 hours where the mice are put into the calorimeter before we actually start analysis of the data, partly because we want them, to get used to the calorimeter, but also get used to their new houses. But unfortunately, at least in this IMPC data set, it looks like most of the runs were actually about 24 hours long, which does not include an acclimation time. So it is possible that if they were given a longer acclimation time, that we would see either differences in the calorimetry output.
And I just had one final question on your data set where you were looking at the age of the mice. Were you perhaps surprised that age didn't contribute more to metabolic rate?
I was somewhat surprised because I think of weight being something that is definitely affected by age. You think that mice are at their heaviest sort of in middle age and then lose weight, they're thinner, younger, and then lose weight as they're older. So I would have thought that at least that difference in body weight would have more of an effect on the metabolic rate. But I mean, comparing the sort of magnitude of differences in energy expenditure among those ages, it is still similar to some genetic differences that we're seeing, metabolic differences that we're seeing among different genes. So it still could be a factor, but it's just, dwarfed by looking at things like the effect of temperature or the adrenergic receptor on, the energy expenditure.
Great, thank you, June. So just had a few questions for Katherine as well. So in the UK Biobank data, does Mpx have differential effects in males versus females?
So yes, I've checked this for most of my gene candidates, and for this particular one, it seems to exert the exact same effect on the two sexes.
Okay, great. And then let me just get back up. So what proportion of the DEXA data in the biobank was missing, and could that affect imputation accuracy?
Mm-hmm. Yeah, so as I said, we only have, when I did this work in 2017, we only had DEXA phenotypes for 5,000 participants out of 500,000. Of course, the more you have, the better your predictions will be. But we saw very good correlations between the imputed and the measured DEXA for most of the phenotypes that I was using. So we were pretty happy about that.
Okay, great. And then, so I think just following on from that is a question from Hannah Taylor from UCL. He says, really interesting talk, just a couple of questions. By doing multiple imputation, you make assumptions that the DEXA data were missing at random and not that they were missing not at random. Can you confirm if that was the case? And as biobank is a convenience sample and not randomized, might this not bias the total allele score for the participants in the cohort?
Mm-hmm. Yes, so DEXA clinics were only carried out for a few of the assessment centers. So all of the participants that had DEXA measured, I believe, fell within 3 or 4 assessment centers. So yes, obviously they weren't evenly sampled throughout the UK. We've tried to correct within our GWAS data for non-random effects. And we've also only used white British participants for this study to try to correct a little bit as well. It was just an issue of what data was available and how could we make the most out of it at the time. So some of these things are out of my control, but a very good question, thank you.
Great, thanks, Catherine. And so there's another question from Philip Langton, who's asking, metalloproteases are implicated in hypertension. We know that obesity and hypertension is correlated in individuals and populations. And do you have any data on blood pressure of the Mpx knockout mice?
Right, so for this particular gene, there hasn't been, there are no GWAS associations with any hypertension or heart disease phenotypes for this gene. I haven't seen anything in my own data. So we're not sure if there is a link for this particular gene and the hypertension phenotype. So we haven't measured it in the mice, no.
Great, thank you. So there's a question from Eleanor Raffan and Fatima from Liverpool John Moores, who both want to know about how MMPX could be working at the molecular level.
Mm-hmm.
Do you have any histology? Would that be sensitive enough to identify differences in adipose structure, development, etc.?
So I briefly showed, I looked at the adipocyte size and number in the gluteofemoral and the gonadal fat pads of the heterozygous and wild-type mice, and I compared them and I saw no differences. But the histology for the homozygous animals was has been delayed due to work restrictions at the moment. So we'll be looking at that, yes. On the hypothetical, from a hypothetical perspective, because this is a new model, we're not entirely sure what to expect. There have been similar models in the literature. It could go either way, basically. So as I said, MMPX affects availability of mature collagen fibers in the extracellular matrix. When adipose tissue expands, it needs collagen to structure itself properly. So having less of them available might constrain the expansion, or it might aid it in a way that we're not sure about at the moment. This is what I'm currently working on to try to identify the in vitro adipose tissue mechanism. So hopefully in a few months, I'll have an answer to this question. Thank you both.
Great, thank you, Catherine. So I just had one. So you showed some gene expression data, I think it was gene expression data, and there seemed to be no difference in brown adipose tissue between the wild type and the knockout, but they exhibit increased energy expenditure. So what do you think could be driving that increased energy expenditure, which I assume is at sort of standard housing conditions rather than anything else?
That wasn't gene expression. It was just organ weights at the postmortem.
Okay, fine.
So it just showed that there were no gross differences in the anatomy. But yes, it's one of the things that I do want to do once I'm able to continue this experimentally, because as you said, a difference in brown adipose tissue would explain the energy expenditure phenotype very easily. So it's one of the things that I need to work on. Okay, great. And then last couple of questions for you, and then one more for June. And we'll finish off there and Joe will close up. So there's a question from Kenneth Verboven. He's asking whether there might be a role for adipose tissue inflammation following the high-fat diet in your MMPX data. Yes, that's one of the hypotheses that I think, I think basically, as I showed in the post-mortem slide, we also saw a decrease in gut length in the homozygous knockout animals, which would corroborate a general inflammation phenotype in them. So this is also, alongside the brown adipose tissue aspect, I think that I'm going to be working on.
Okay, excellent. And then, so a question from Laura Oller, who thanks for both for great presentations. And in your opinion, if you were to compare results from different species, i.e. humans, rats, mice, for example, which adiposity parameter would you use, if any, to do that?
I think it depends entirely on what your scientific question is. As I discussed before, they're all better suited for different things. I bashed BMI a little bit, but of course, if you're looking at big population data, it is useful to a certain extent. So it just depends on what you're trying to ask and what you're trying to show.
Great, thank you. As always, context, terribly important. And then final question for June. So it's a question on food intake, and do you use body weight as a covariate when you're analyzing food intake data?
Yes, we do use body weight as a covariate for food intake. We use, well, to be clear, depending on the experimental question, we will use either total body weight or lean body mass, usually as a covariate for most of the metabolic parameters, including food intake. Generally, the only metabolic parameters that we don't use body weight or body composition as a covariate is generally respiratory exchange ratio, which is a ratio and not dependent on body weight, as well as ambulatory activity.